Hello everyone, today we are going to talk about the chapter excretory products and its elimination, out of which a key concept that is about physiology of urine formation. आज हम वो डिस्कस करने जा रहे हैं. Human being belonging to class Mammalia, we are called as ureotelic animals. और जब हम यूरियोटेलिक बोलते हैं यानी कि हमारे बॉडी का चीफ नाइट्रोजनस वेस्ट जो हम यूरिन के थ्रू एलिमिनेट करने वाले हैं, दैट इज यूरिया. So human body does not have any ability to store excess of amino acid. हम हमारे बॉडी में कार्बोहाइड्रेट, एक्सेस ऑफ कार्बोहाइड्रेट यानी कि ग्लूकोज को ग्लाइकोजन के फॉर्म में स्टोर करते हैं, दैट इज इन आवर मसल्स एंड इन आवर लीवर. हमारे बॉडी में फैट को भी, एक्सेस ऑफ फैट को भी स्टोर करने के लिए हमारे पास एडिपोज टिश्यू है, बट हमारे बॉडी में एक्सेस ऑफ अमीनो एसिड को स्टोर करने के लिए कोई भी स्ट्रक्चर नहीं है. And each and every amino acid which is in excess gets deaminated, that is getting converted into ammonia, and that ammonia gets converted into urea. Deamination more or less हमारे बॉडी के हर सेल में होता है, बट जब हम अमोनिया टू यूरिया की बात करते हैं, that happens only and only in liver cells. Liver is the only organ which has an ability to convert ammonia into urea. As we know, ammonia which is highly toxic and it has to be eliminated as soon as it has been formed, or it has to be converted into less toxic substance. So let us understand the nitty-gritty of the same in detail. So now when we talk about physiology of urine formation, we divide it into two parts. That is chemical part of this physiology and the mechanical part. Under this chemical part, we will talk about how ammonia is getting converted into urea, or I would call it as a urea cycle. Urea cycle, which we also call it as a ornithine cycle, or we also call it as a Krebs-Henle cycle. So we will be talking about this particular process, biochemical process. After that, urea is carried to the kidney. So first, liver comes into play for the conversion of ammonia into urea, and then that urea is carried to the kidney, where mechanical part of this urine formation takes place, and which we divided into three headers. Number one, glomerular filtration, which we also call it as ultrafiltration. Number two, selective reabsorption. And number three, tubular secretion. So let us understand each of this process in detail, starting with the first, urea cycle or conversion of ammonia into less toxic urea. So as we said, this process occurs only and only in the liver cells, or what we call it as the hepatocytes. So हमारे बॉडी में जितना भी अमोनिया है, that is all brought to this hepatocytes, and hepatocyte के साइटोप्लाज्म और माइटोकॉन्ड्रिया के अंदर ये प्रोसेस होती है. If we look at its steps, the first, that excess of amino acids which enters into the mitochondria, where one molecule of ammonia along with the carbon dioxide using the energy gets converted into carbamoyl phosphate. So this step of this urea cycle, the very first step of the urea cycle taking place in the mitochondria of the hepatocyte, where ammonia combining with carbon dioxide forms carbamoyl phosphate, and that carbamoyl phosphate binds with ornithine. Ornithine is one of the amino acid. This is my non-protein amino acid. यानी कि ऑर्निथिन ऐसा अमीनो एसिड है जो कोई भी प्रोटीन चेन में आपको नहीं मिलेगा, बट वेरी मच रिक्वायर्ड फॉर सच बायोकेमिकल प्रोसेसेस इन आवर बॉडी. And now this ornithine, which is combined with carbamoyl phosphate, forms citrulline. Citrulline getting converted into arginosuccinate with the addition of another amino group coming from the cytoplasm of the hepatocyte. और यहाँ पे वो arginosuccinate split होता है, giving rise to arginine and fumarate or fumaric acid. And then this arginine acted upon by the enzyme arginase give rise to urea and reforming ornithine for picking up a next carbamoyl phosphate. And that's the reason we call this as ornithine cycle or the end product, which is urea cycle, or the name from the founder, that is Krebs-Henle cycle, where two molecule of ammonia, one which is getting added at this step in the mitochondria, and second which is getting added in the cytoplasm, getting combined with one molecule of carbon dioxide, forming urea. So this is the cycle which takes place only in liver cell, and that's why it is said that अगर आपका liver damage होता है, if you suffer from a liver cirrhosis or a failure, आपके बॉडी में अमोनिया का लेवल बढ़ जाएगा. किडनी फेल होता है तो यूरिया बाहर नहीं निकलेगा, बट अगर लीवर फेल होता है, अमोनिया लेवल विल इंक्रीज, एंड वंस द अमोनिया विच इज अ हाईली टॉक्सिक सब्सटेंस, अगर उसका लेवल इंक्रीज होता है, डेफिनेटली इट्स लाइफ थ्रेटनिंग. So now this urea which has been formed by the liver gets carried to the kidney for its elimination. So that's our next discussion, that how, or from liver, how it is getting transported to the kidney. So as we said, urea is being formed by liver cells. So from liver, the first blood vessel which carries urea is my hepatic vein. I repeat, from liver, hepatic vein is the first one to carry all this urea to the inferior vena cava, from where it goes to the right side of the heart, from where it goes to the pulmonary artery, reaching to the left side of the heart. From here, it goes to the aorta, and aorta, that I would say dorsal aorta precisely, carries it to the renal artery, or dorsal aorta, which divides into renal artery, and through renal artery it reaches to the kidney, where all this urea is going to be filtered out, and the blood which is coming out from the kidney, that is via renal vein, back to the heart, right? So it is said that hepatic vein is the one which contains highest amount of urea, or you can say maximum amount of urea, it will be found in hepatic vein, whereas the renal vein is the vessel which has the least amount of urea. MCQ point. The maximum amount of urea will not be in the renal artery. You may get confused that renal artery is the one which is carrying it to the kidney, तो उसमें सबसे ज्यादा होगा यूरिया, but no, just understand, लीवर से वो सबसे पहले कहाँ जा रहा है, तो सारा यूरिया किसमें पोर आउट हुआ, हेपेटिक वेन में, उसके बाद वो किडनी तक पहुंच रहा है, उस दौरान it gets distributed along the network of blood vessels, but yes, here it is maximum, and then किडनी में यूरिया फिल्टर आउट होने के बाद जो ब्लड बाहर आ रहा है, that is the blood present in the renal vein, will have a least amount of urea. So that was all with respect to chemical part of the urine formation and its transport to the kidney. Next, that is about mechanical part, that what role kidney plays, or how kidney eliminates the nitrogenous waste along with other waste product from our body. So now when we look at the mechanical part of the urine formation, physiology of urine formation, which plays completely in our kidney, the first step in this process, that is what we call it as glomerular filtration, or we also call it as ultrafiltration. Why do we call it as ultrafiltration? Let us understand that. So the structure of Malpighian corpuscle or a renal corpuscle, which we studied earlier, where we have this double layered Bowman's capsule, which contains a network of capillaries, what we call it as glomerulus, afferent arteriole, which carries the blood towards the glomerulus and the efferent arteriole which drains out the filtered plasma, filtered blood away from the glomerulus. So जो भी filtration होना है which is taking place in this glomerulus and the Bowman's capsule, what are the factors and how does it take place? So if you recollect, in this inner membrane of the Bowman's capsule, we have a specialized cells which we call it as podocyte, a cells with a feet-like process, or what we call it as slit-like processes. So obviously they are going to allow only selected molecules from the blood, from the glomerulus to get filtered out, यानी कि वो molecule जो इसके pores यानी कि इसके slit से pass हो पाएंगे, only they will be coming in the cavity of the Bowman's capsule. बाकी का कोई भी substance यहाँ पे filter out नहीं हो सकता, right? So the molecule which has a size larger than the slits won't be able to pass through this. Okay, that's one point. Now, what is leading to this filtration to take place? Let us understand that. So, to understand the process of this glomerular filtration, we need to understand three important pressure. The first one, which we call it as GHP, that is glomerular hydrostatic pressure. Second, we call it as BCOP, that is blood colloidal osmotic pressure. And third, we call it as CHP, that is capsular hydrostatic pressure. So let us understand each of this one by one. Now when I say GHP, glomerular hydrostatic pressure, which has been created due to the difference in the diameter of afferent and efferent arteriole. Understand this. Afferent arteriole, which has a more diameter, which is wider than the efferent arteriole, which is comparatively narrower. यानी कि यहाँ पे जो ब्लड आ रहा है और यहाँ से जो ब्लड ड्रेन आउट हो रहा है, उनके बीच में अगर डायमीटर में डिफरेंस है, वो पाइपलाइन के डायमीटर में डिफरेंस है, ऑब्वियसली देयर विल बी प्रेशर क्रिएटेड इन द ग्लोमेरुलस. यहां से ज्यादा ब्लड आ रहा है और यहां से कम ब्लड बाहर जा रहा है, सो ऑब्वियसली द टफ्ट ऑफ कैपिलरीज, दैट इज ग्लोमेरुलस, विल बी अंडर प्रेशर, एंड दैट इज व्हाट वी कॉल इट एज GHP, glomerular hydrostatic pressure. As I said, this is due to difference in the diameter of afferent and efferent arteriole. So as we said, afferent arteriole are wider than the efferent. This is created. Second, BCOP, blood colloidal osmotic pressure. If you recollect from the composition of blood, we come across the important protein molecule which is present in our blood, which maintains the blood colloidal pressure, and that is albumin. So such large protein, binding protein, does not allow the component of blood to get leaked out, and that creates my blood colloidal osmotic pressure, or some books says as a blood colloidal oncotic pressure. So this BCOP, which is going to act against the filtration to take place, because वो मेरे ब्लड के कंपोनेंट को बांध के रखेगा, बाइंड करके रखेगा, वो फिल्टर आउट नहीं होने देगा. So that BCOP, which is mainly due to proteins, and which protein precisely, if I have to say, then that is albumin. And third, that is CHP, that is capsular hydrostatic pressure. अब जब यहाँ पे filtration हो रहा है, some fluid will always be collected over here, which will also be resisting the filtration to take place, and that will be called as capsular hydrostatic pressure. So this is basically due to filtrate present in Bowman's capsule, and that we call it as capsular hydrostatic pressure. So three pressure we talked about here. One, GHP, that is due to the difference in the diameter of afferent and efferent arteriole. Second, BCOP, that is because of the protein albumin, which is against the filtration to take place. And third, CHP, that is because of the fluid present or a filtrate present in the capsule, which is resisting the filtration to take place. So now when I look at the net filtration pressure, that's a difference of this three. So my net filtration pressure, that's the difference between this three, is about 10 to 20 mm of Hg. So how do we calculate? It is said that my glomerular hydrostatic pressure, which is about 55 to 70 mm of mercury, whereas my blood colloidal osmotic pressure is about 30 mm of mercury, and my capsular hydrostatic pressure, which is about 15 mm of mercury, right? So glomerular hydrostatic pressure was in favor of filtration to take place, whereas these two are opposing the filtration. So obviously my net filtration pressure or effective filtration pressure is going to be GHP minus BCOP minus CHP. So that is about 55 minus 30 minus 15. That is about 10 mm of mercury. Right? Some book gives it at 70 as GHP, so that ranges from around 10 to 20 mm of mercury. So your MCQ point, your net filtration pressure will always be in the range of 10 to 20 mm of mercury, and that's why this process we also call it as ultrafiltration. This process we also call it as ultrafiltration. Now the question is, how much filtrate is produced? So it is said that per minute around 20 to 25% of total cardiac output reaches to kidney. मतलब अगर हम कैलकुलेट करने जाते हैं, the cardiac output which is about 5 liters, उसका 20 से 25% blood comes to kidney. Okay? So अगर मुझे इससे कैलकुलेट करना है तो I would say about 1000 to 1200 ml of blood comes to kidney. और उस 1200 ml में से every minute 125 ml gets filtered out or filtrate is formed. Out of 1200 ml per minute, कितना filtrate बन रहा है? 125 ml. So this is what I call it as GFR, that is glomerular filtration rate. This is what we call it as glomerular filtration rate. So per minute 125 ml. Obviously this comes to about 7.5 liter per hour, or I would say 180 liter per day. Every day 180 liter filtrate बनता है. Every hour around 7.5 liter, or every minute about 125 ml filtrate बनता है. Also understand over here, अगर मेरा ब्लड प्रेशर, मेरा सिस्टमैटिक और सिस्टमिक ब्लड प्रेशर अगर चेंज होता है, ऑटोमेटिकली मेरा नेट फिल्ट्रेशन प्रेशर भी चेंज होगा. द मोमेंट माई सिस्टमिक ब्लड प्रेशर इज चेंज, मेरा जीएचपी चेंज होगा. यानी कि अगर मेरा ब्लड प्रेशर कम हो जाता है, मेरा जीएचपी कम हो जाता है, by default its effect will be seen on NFP, that is my net filtration pressure. If this reduces from 55 to 70 से लेके मैं उसको बोल दूं ये हो गया मेरा 45 से 50, तो obviously my net filtration pressure will be 0. In that case urine formation will be affected. That's why it is said अगर आप hypotension यानी कि आप low blood pressure से भी suffer कर रहे हो, then it is going to be harmful, right? Because it is going to ultimately affect your NFP. And that will affect your urine formation. आपके body से nitrogenous waste, metabolic waste बाहर नहीं जाएंगे. So that's why maintaining blood pressure is very, very important, right? So over here, when we talk about this GFR, glomerular filtration rate, the filtrate which has been formed, which has been collected in our Bowman's capsule, what is its composition? At the same time, there is one very important terminology to understand, and that is what we call it as a filtration fraction. So when I talk about filtration fraction, that is GFR upon renal plasma flow पर मिनट कितना फिल्ट्रेट बना अपॉन पर मिनट कितना प्लाज्मा किडनी के पास गया. सो इफ यू रिकलेक्ट योर डेटा 1200 ml में से अगर मुझे प्लाज्मा का परसेंटेज निकालना हो, दैट इज 55%. हमारे ब्लड का 55% इज प्लाज्मा. सो ऑब्वियसली 1200 का 55 वुड बी समवेयर क्लोज टू 650 ml. एंड एज वी सेड जीएफआर कितना है 125. सो दिस कम्स टू अबाउट 0.19, या अगर मैं उसको परसेंटेज में कैलकुलेट करता हूं 19 to 20%. That is my filtration fraction. That is amount of plasma going out of which filtrate formed. So these are very key important MCQ point. Coming back to the point, what is the composition of filtrate? यानी कि हमारे ब्लड में से क्या क्या कंपोनेंट फिल्टर आउट हुए, and what is the next step? So that's with respect to glomerular filtration or ultrafiltration. Next, let's talk about selective reabsorption. But before that, first, what are the composition of glomerular filtrate? So now when we talk about the composition of glomerular filtrate, it has been said that all components of plasma gets filtered out except proteins, because proteins having a large size cannot pass through those slits of the podocyte. That's why they do not get filtered out. But बाकी का जितना भी composition हमारे प्लाज्मा का है, प्लाज्मा, I am not talking about blood, प्लाज्मा का है, that all gets filtered out. And that's the reason glomerular filtrate can also be called as deproteinized plasma. We can also call it as deproteinized plasma, because plasma में से प्रोटीन निकाल दो, बाकी का सारा फिल्टर आउट हो गया. Now the point is it okay to get all this waste or all this filtered molecule to be eliminated? The answer is definitely no. It is said that 99% of the filtrate, because simple सा logic है, अगर 180 liter filtrate बनता है दिन में, do we urinate that much? Obviously not. We urinate about 1 to 2 liter depending on the physiological condition of a body, depending on the climatic condition, depending upon the water intake, and multiple other factors. But yes, more or less 99% of the filtrate formed is reabsorbed. Okay, so we divide these components of the glomerular filtrate into 3 categories. So these 3 categories are: number 1, high threshold substances; number 2, low threshold substances; and number 3, no threshold substances. यानी कि वो substances, high threshold substances are those substances which are reabsorbed completely. इनको मैं एलिमिनेट होने नहीं दे सकता, ये मुझे वापस हमारे ब्लड में चाहिए. एंड दे आर मेनली ग्लूकोज, sodium, along with some essential amino acids. They are my high threshold substances. I cannot afford to lose them. Low threshold substances are those substances which are reabsorbed in small quantity. because they are required to maintain the homeostasis, and they are less toxic substances such as urea and uric acid. These are my low threshold substances. And when we talk about no threshold substances, यानी कि ये substance किसी भी कीमत पे should not be reabsorbed because they are so toxic, they need to be eliminated completely. No threshold substances need to be eliminated completely. मतलब उनको एलिमिनेट करने के लिए अगर मुझे एनर्जी भी स्पेंड करनी पड़े, जैसे हाई थ्रेशोल्ड सब्सटेंसेस को रीएब्सॉर्ब करने के लिए मुझे एनर्जी स्पेंड करनी पड़ेगी, उसी तरह नो थ्रेशोल्ड सब्सटेंसेस टू एलिमिनेट देम इवन इफ आई नीड टू स्पेंड एनर्जी, दैट्स फाइन, एंड they are basically my highly toxic substances such as ammonia or creatinine or hippuric acid or any drug substance which has been consumed for anything. So these are my no threshold substances, should be eliminated completely. So let us understand how this selective absorption followed by elimination occurs in our body. That's our next step. Mechanical process ki do step: selective absorption or tubular reabsorption and tubular secretion. So now when we talk about selective reabsorption या फिर tubular reabsorption, we are using the word reabsorption because ये न्यूट्रिएंट जब हमने कंज्यूम किया था, जब हमने डाइट में लिया था, तब पहली बार इंटेस्टाइन ने उनको एब्जॉर्ब किया था, ब्लड का पार्ट बनाया था. अब ये ब्लड से वापस फिल्टर आउट होके बाहर जा रहे हैं, तो उनको वापस से लाना है. दैट्स व्हाई वी आर यूजिंग द टर्म एज ट्यूबुलर रीएब्जॉर्प्शन, एंड दिस प्रोसेस इज अकरिंग इन रीनल ट्यूब्यूल्स. सो द वर्ड गिवन इज ट्यूबुलर रीएब्जॉर्प्शन. सो इफ वी रिकलेक्ट, ये हमारा बोमेन्स कैप्सूल और वहां से जो हमारी पहली ट्यूब्यूल आती है, दैट इज प्रॉक्सिमल कॉन्वोल्यूटेड ट्यूब्यूल. प्रॉक्सिमल क्लोजर टू द माल्पीजियन बॉडी, coiled like convoluted tubule, and the lining of the same, as we know, it is a brush bordered cuboidal epithelium. Obviously cuboidal होने का बेनिफिट माइटोकॉन्ड्रिया का अबंडेंस एक्टिव रीएब्जॉर्प्शन में हेल्प करेगा. माइक्रोविलाई ब्रश बॉर्डर होने का बेनिफिट having those processes to increase the surface area for reabsorption. It is said that tubular reabsorption का major part, that is 75%, occurs in PCT itself. Maximum reabsorption हमारा PCT में ही हो जाता है. You may get a question which might confuse you. Water का maximum reabsorption कहाँ होता है? PCT, DCT, loop of Henle. And as an instinct, अरे हाँ, loop of Henle में हमारा water, because loop of Henle तो सिर्फ water के लिए specialize बना है, तो वही आंसर होगा. You are making a mistake. 70% of the water reabsorption also take place in PCT. Right? जो हमने यहां बात की थी, 180 लीटर जो हमारा फिल्ट्रेट बना, उसका 99% हमें रीएब्सॉर्ब करना है, और वो 99% में से 70% रीएब्सॉर्प्शन PCT में ही होता है. So now when we talk about the process of reabsorption in PCT, as we said, PCT has this lining of cuboidal epithelium with brush border, यानी कि वो एक्टिवली सब्सटेंसेस को रीएब्सॉर्ब करेगा और कहां डालेगा? ब्लड में. दैट मींस द सेल्स ऑफ PCT will reabsorb high threshold substances like glucose or sodium from the filtrate into the blood. अब यहां पे मुझे एनर्जी क्यों लगनी है? Because when the filtrate is entering into the PCT, it is said to be isotonic to the blood. जब फिल्ट्रेट बोमेन्स कैप्सूल से PCT में जा रहा है, इट इज आइसोटोनिक टू द ब्लड, यानी कि दोनों की ऑस्मोलारिटी जब सेम है, और फिर भी अगर मुझे चाहिए कि यहां से सॉल्यूट यहां जाए, यहां से ग्लूकोज या फिर सोडियम फिल्ट्रेट से ब्लड में जाए, आई एम गोइंग टू वर्क अगेंस्ट द कॉन्सेंट्रेशन ग्रेडियंट, एंड डेफिनेटली ओवर हियर ATP will be used, energy will be used. and by using the energy, I'm taking the solutes from the filtrate into the blood. By default, blood की osmolarity बढ़ गई, filtrate की कम हो गई, water passively move करेगा from the filtrate into the blood. And this kind of water reabsorption, which is occurring passively, is called as obligatory water reabsorption. Because उसको obligation है, obligation to maintain the osmolarity. पीसीटी के सेल्स ने हाई थ्रेशोल्ड सब्सटेंसेस को एक्टिवली रीएब्सॉर्ब किया, ऑस्मोलारिटी मेरी अफेक्ट हुई, वाटर विल फॉलो. सिमिलरली certain bicarbonate ion will also diffuse, right? So these are the major reabsorption steps which are taking place in PCT. Via this 100%, I repeat, 100% glucose and up to 70 to 80% sodium as well as potassium as well as bicarbonate are getting reabsorbed along with water in PCT itself. That's why we say overall 75% of my tubular reabsorption is going to take place in PCT. So basically the filtrate which entered into the PCT was isotonic to the blood, and now the filtrate which is leaving the PCT will still remain isotonic to the blood. The reason जितना solute reabsorb हुआ उतना water भी तो reabsorb हुआ. So I repeat, the filtrate entering and leaving PCT is said to be isotonic to the blood. Is it clear? That's why we say that PCT plays a very important role in maintaining pH of our blood as well as ionic composition of our blood. PCT plays a very important role, right? Next, we see what happens in loop of Henle and then reabsorption in DCT. Talk about reabsorption in loop of Henle or Henle's loop. As we know, we have two limbs of loop of Henle. That is descending limb and the ascending limb. First, if I look at the important MCQ point, descending limb, which is lined by simple squamous epithelium, is said to be permeable only to the water and impermeable to the electrolyte. Whereas ascending limb, जहां पे minimum reabsorption होता है, is said to be permeable to the electrolyte and impermeable to the water. Please understand, descending limb, squamous epithelium. Ascending limb, as we know, this is cuboidal epithelium. अब again, cuboidal epithelium है, so active reabsorption possible है, वो actively electrolyte को reabsorb करता है, so जिसकी वजह से मेरे medullary intestine का किडनी के अंदर का जो interstitial space है, वहां पे electrolyte आने की वजह से osmolarity increase हुआ, and that will drain out water from the descending limb. So what happens is, let's say this is my descending and ascending limb of loop of Henle. This is descending limb, this is ascending limb. Filtrate which is coming out from the PCT, which is said to be isotonic to the blood, entering into the descending limb. Whereas over here, filtrate is moving in upward direction because it is ascending limb. These are my cuboidal epithelium, and this simple cuboidal epithelium reabsorbs solute, or I would say electrolytes. ये electrolyte को reabsorb करेगा, मेरा medullary interstitium का osmolarity increase होगा बाय डिफॉल्ट यहां से वाटर ड्रेन आउट होगा. So when the filtrate is losing out the water to the medullary interstitium, osmolarity will keep increasing, and when it reaches to this hairpin band, it is a maximum osmolarity. The filtrate has a maximum osmolarity when it reaches to the hairpin band. And now when it is moving upward, यहां से water तो नहीं जाएगा, but yes, electrolyte जा रहा है. So as the electrolytes are moving out, osmolarity will decrease. That means it will again, the filtrate will again become diluted, or at times it is the hypotonic before entering into the DCT. So in loop of Henle, though we say that water and electrolyte are getting reabsorbed, but understand, हमारे बॉडी में, हमारे किडनी में, ह्यूमन किडनी में only 15 to 20% nephron are juxtamedullary nephron. बाकी के 80 to 85% are cortical nephron, right? In anatomy of this excretory system, when we discuss about types of nephron, we come across this. So ये जो mechanism है, जिसको हम counter current mechanism बोलते हैं, जिसको हम अलग से discuss करेंगे, then counter current mechanism is getting played only in 15 to 20% of the nephron, which are helping us to concentrate the urine. So this is how descending limb of the loop of Henle allows the water to be reabsorbed, provided interstitial medullary osmolarity is increased by the active reabsorption by ascending limb of loop of Henle. Now when we talk about reabsorption in DCT-CT, this is purely conditional reabsorption which takes place under the action of hormones. I repeat, in DCT-CT, reabsorption is conditional under the action of hormones. यानी कि जब मैं conditions की बात कर रहा हूं, physiological condition of our body will determine what reabsorption or which molecule reabsorption does take place, and that occurs via hormone. For example, if our body has lost quite a lot of solutes, or I would say salt, in the form of perspiration, अगर हमें बहुत ज्यादा तेज धूप है और पसीना हो रहा है और हम salt और water को lose कर रहे हैं, then definitely our body releases, I mean to say our adrenal cortex releases aldosterone, or what we also call it as mineralocorticoid, और ये हमारे बॉडी में, ये हमारे डीसीटी पे एक्ट करके सोडियम एंड वाटर को रीअब्सॉर्ब करेगा. ऑब्वियसली इन रिटर्न ये पोटेशियम और H+ को रिलीज करेगा, यानी कि एक्सक्रीट, या फिर आप बोल दो सिक्रीट करेगा. और एल्स अगर हमें सिर्फ वाटर रीअब्सॉर्ब करना है, देन ADH, एंटी डायूरेटिक हार्मोन, या फिर हम उसको हम वेसोप्रेसिन भी बोलते हैं, दैट विल एक्ट ऑन DCT एंड रीअब्सॉर्ब वाटर. या फिर आप एक और हार्मोन दैट इज PTH, पैराथायरॉइड हार्मोन, और पैराथ हार्मोन ये हमारे DCT से कैल्शियम को रीअब्सॉर्ब करेगा. यानी कि ये सारे स्टेप्स हमारे फिजियोलॉजिकल कंडीशन पे डिपेंड करते हैं, बॉडी को किस पर्टिकुलर चीज की रिक्वायरमेंट है. एंड दैट्स द रीजन यहां पे जो हम वाटर रीअब्सॉर्प्शन की बात करते हैं, दैट वाटर रीअब्सॉर्प्शन इज कॉल्ड एज फैकल्टेटिव रीअब्सॉर्प्शन. जो हमारा वाटर PCT में रीअब्सॉर्ब हुआ था, वी कॉल्ड इट एज ऑब्लिगेटरी वाटर रीअब्सॉर्प्शन. Over here we call it as facultative water reabsorption. So basically if I talk about this DCT, अगर हम DCT की बात कर रहे हैं, DCT के लेवल पे सोडियम रीअब्सॉर्ब होगा, वाटर रीअब्सॉर्ब होगा, even some amount of bicarbonate can be reabsorbed, but against this sodium potassium and H+ will be excreting. So that's like an antipode mechanism. Or even in special condition, calcium under the action of PTH will be reabsorbed. यहां पे हमारा हार्मोन आएगा aldosterone and ADH, whereas this will also be eliminated under the action of aldosterone to get the sodium into the blood to maintain blood pressure and blood volume. So that's about reabsorption in DCT. That's our second step of the urine formation or the mechanical part, that is selective reabsorption. So अगर मैं उसको quickly recap लेता हूं इस particular part का. So yes, as we discussed, at the level of PCT, again this diagram based question were asked in 2025, very important part. At the level of PCT, all this molecule are coming from the filtrate into the blood. That is about reabsorption. You might get confused. Sir, यहाँ आप बोल रहे हो पोटेशियम को reabsorb किया और DCT के लेवल पे पोटेशियम को secrete किया, eliminate किया. Then understand the difference. Over here, that is happening based on the requirement of the blood, body. उतना ही पोटेशियम आएगा. और यहाँ पे अगर मुझे हार्मोन अल्डोस्टेरोन के अंदर सोडियम को लाना है तो पोटेशियम एंड H+ एलिमिनेट करना पड़ेगा, राइट? देन इन limb of loop of Henle, that is descending limb, water is coming, ascending limb, electrolytes are coming. Again, descending limb permeable only to water, impermeable to solute. Ascending limb permeable only to solute, यानी कि electrolyte impermeable to water. Whereas in DCT level, you can see NaCl, water or even bicarbonate comes under the action of hormone, against which potassium and H+ are going out. Whereas collecting duct, यहाँ पे आपका water reabsorption हो रहा है, but at what level? In the inner medullary interstitium, because of the higher medullary osmolarity, water is getting reabsorbed and then आपका urine hypertonic form में eliminate हो रहा है. We are yet to talk about the third step, that is tubular secretion. Let us understand that first. So now when we talk about tubular secretion, obviously it occurs at two important locations in our nephron, that is at the level of PCT and number 2 at the level of DCT. सर हमने सारी चीजें फिल्टर आउट तो कर दी थी, अब हमें सिक्रीट क्या करना है? So as we discussed earlier कि जो हमारा लो थ्रेशोल्ड सब्सटेंस है, जो हमारा लो थ्रेशोल्ड सब्सटेंस है, अगर वो किसी भी क्वांटिटी में हमारे ब्लड में रह जाता है, then it is going to be definitely harmful for us, right? So हमारे लो थ्रेशोल्ड सब्सटेंस को हम पीसीटी के लेवल पे, आई रिपीट, हमारे नो थ्रेशोल्ड सब्सटेंस को पीसीटी के लेवल पे हम एलिमिनेट करते हैं. We eliminate it even by spending energy. So that's why we also call it as tubular secretion. ब्लड से फिल्ट्रेट में जाना. अब तक जो हमने बात की वो फिल्ट्रेट से ब्लड में लाया था, अब यहां जो बात कर रहे हैं उसको ब्लड से फिल्ट्रेट में डालना. So that is happening at the PCT level, and even at the DCT level, when we are reabsorbing sodium, we need to eliminate excess of K+ and H+. So these are the two locations where tubular secretion will also take place to maintain the pH and ionic balance of our body fluids. So that's about the process of physiology of urine formation.